Okay. Good morning, students. After 1 week after the unit test, now we are going to see the lessons again. So I have started lesson 5, molecular genetics. We have discussed about the molecular genetics. Genetics. What is the meaning of genetics? Study about the genes are known as genetics. Where the genes are located inside the chromosome. Where is chromosome is located? Inside the DNA. Inside the DNA nucleus, these chromosomes are located. So now we are going to discussing. Now we are discussing the genes. So gene is the basic physical and functional unit of heredity. Then few characteristics of genes. What are the properties of the genes we have discussed. Then we have seen what? Genetic material. Which one is the genetic material? DNA or RNA or protein? Which one is the genetic material? So in mostly all the organisms, DNA is the genetic material. In some of the viruses, RNA is a genetic material, not protein. Okay. Then the protein, it is essential for a number of biochemical reactions. Okay, number of biochemical reactions are occur in our body. That reactions need to occur proper. We need the protein. But protein is not a genetic material. Then what we have discussed, then that Avery et al. Experiment we have discussed about the DNA as a genetic material. Like that same Hersey and Hayes experiment also we have discussed about DNA is the genetic material. Then the DNA, it is present mostly two types of DNAs. Okay, what DNA it is contains, it is made up of a nucleic acid. Okay, so two types of nucleic acid, deoxyribonucleic acid and ribonucleic acid. Then we have seen, so that nucleic acid, chemical structure of nucleic acid, it is present in the nucleotide. Repeated subunits of the nucleotides are present. That nucleotides, it is present in the three parts: nitrogenous base, 5-carbon sugar and phosphate group. That we have discussed. Okay, sugar nrappa deoxyribose sugar and ribose sugar. That we have seen. The nitrogenous base in DNA and RNA, it is present 4 nitrogenous base. The 2 is the pyrimidine, 2 is the purine. Then that here adenine and guanine is the bases have a double carbon bond. So that thymine and cytosine, uracil, it is a pyrimidine, it is present in the uracil, it is present only in the RNA. Thymine is present only in the DNA. That is the difference between DNA and RNA. That unique structures we have seen. So 2 purines and 2 pyrimidines we have seen. Okay, adenine and guanine is the purine and thymine and cytosine is the pyrimidine. Then phosphate is the functional group of a nucleic acid. Okay, so phosphate group is the functional unit of nucleic acid. That we have seen. Then we have seen what? Nucleoside and nucleotide. Okay, nucleoside and nucleotide we have seen. So that nitrogenous base is linked with the one molecule of sugar. It is forming in a what? Nucleoside. Then in the nucleoside, phosphate group, it is attached at the 5 dash of carbon. That is known as the nucleotide. That we have discussed. Nucleoside and nucleotide. Then that more points, everything we have discussed. Then RNA world, that also we have discussed. Then what are the properties of genetic material? So genetic material have certain properties. What are the properties? Cell replication, stability, information storage, then variation through the mutation. That we have seen. Then we have discussed about the packaging of DNA helix, because DNA, it is arranged in a linear manner. Okay, it is present in the number of in a length of the double helix DNA model, it is present, but it is packed inside a small nucleus. How it is packed, that we have discussed. Okay, so the DNA, it is presence in the histone proteins. That histone proteins bonding the DNA. Okay, number of polynucleotides are present. That polynucleotides are present neighbor to the DNA structure. This histone protein, it is bond the two molecules of a histone proteins. Okay, it is bonding with that, then it is producing in a like this structure. Okay, so in a larger length of this nucleotides are located inside the small nucleus. Packaging, how the DNA it is packed inside the nucleus, that also we have discussed everything. Once you must recall. Okay, all these fully you must once recall, you read again and again, then only you will possible to understand this lesson. Okay, the next now we are going to discuss about the DNA replication of a DNA. So replication, already I have said, multiplication. The same copies they can produce. The DNA, they can produce the same copies of the DNA in a fast. Okay, that process is known as the replication. So replication of DNA takes place during the S phase of cell cycle. Cell division. Okay, so the cell, single cell, it is divided into two. That is known as the cell cycle. That cell cycle, it is present in the number of phases. One of the phase is known as the S phase. So the S phase of the cell cycle, DNA, it is involving in the process of replication. During replication, each DNA molecules give rise to two DNA strands identical to each other as well as to the parent strand. So the replication process, DNA molecules, they can produce the same DNA copies. The DNA molecule, it is producing in a same identical copies, or then it is producing in the parent strand. Parent strand எப்படி இருக்கோ அதே மாதிரி ஒரு new offsprings it is producing, or we can say it is producing in the identical copies. That process is known as the replication. Then three hypotheses of DNA replication has been proposed. So they have identified in a three methods, three ways of the DNA replication. So one is known as conservative replication, then dispersive replication, and semi-conservative replication. So DNA replication, DNA, it is involving to produce the exact carbon copy of a same molecule of DNA. That is known as the DNA replication. It is present in the three methods. Okay, conservative method, dispersive method, and semi-conservative method. Okay, first one is the conservative replication. The original double helix serves as the template. இங்கே என்ன இருக்கும், இப்போ ஃபார் எக்ஸாம்பிள் இதுதான் டிஎன்ஏனுடைய ஸ்ட்ரக்சர், டபுள் ஹெலிக்ஸ் ஸ்ட்ரக்சர் அப்படின்னு சொல்லிட்டோம்னா, இந்த டிஎன்ஏ டபுள் ஹெலிக்ஸ் ஸ்ட்ரக்சர் இன்னும் கன்சர்வேட்டிவ் மெத்தட்ஸில் அப்படியே இதே மாதிரி இன்னொரு காப்பீஸை ப்ரொடியூஸ் பண்ணும். தட் மெத்தட் இஸ் நோன் அஸ் த கன்சர்வேட்டிவ் மெத்தட். டெம்ப்ளேட்னா என்ன, அதுக்கும் சேம் மீனிங் தான், ரெப்ளிகேஷன்ற மீனிங் தான் டெம்ப்ளேட் அப்படின்றது. அதே மாதிரி உள்ள ஒரு காப்பீஸை அது ப்ரொடியூஸ் பண்ணுது. Okay, so the original molecule is preserved intact and an entirely new double stranded molecule is synthesized. So original இருக்கக்கூடிய அந்த parent molecule அப்படியே preserve intactனா என்னது, அது எந்த ஒரு ஆக்டிவேட் இல்லாமல் அது மாறிட்டு என்டையராக அதே மாதிரி இருக்கக்கூடிய ஒரு நியூ டபுள் ஸ்டாண்ட் மாலிகூல்ஸ் வந்து ப்ரொடியூஸ் ஆகும். தட் மெத்தட் ஆஃப் ரெப்ளிகேஷன் இஸ் known as conservative replication. Another one is known as dispersive replication. The dispersive replication, the original DNA molecule is broken into fragments. என்ன செய்யுது, நம்பர் ஆஃப் ஃப்ராக்மெண்ட்டாக broken down ஆகி ஒவ்வொரு ஃப்ராக்மெண்ட்டும் அதே மாதிரி உள்ள இன்னொரு ஃப்ராக்மெண்ட்டை ப்ரொடியூஸ் பண்ணும். Okay, so each fragment serves as a template for the synthesis of complementary fragment. எந்த மாதிரி உடஞ்சிருக்கோ ஃப்ராக்மெண்ட்டாக இருக்கோ அதே மாதிரி உள்ள ஒரு டெம்ப்ளேட் ஃப்ராக்மெண்ட்டை ப்ரொடியூஸ் பண்ணுறது தான் என்ன மெத்தடு, தென் dispersive method. Okay, finally two new molecules are formed which consists of both old and new. ஒரு ஃப்ராக்மெண்ட், ஓல்டு ஃப்ராக்மெண்ட், அந்த ஓல்டு ஃப்ராக்மெண்ட் அதே மாதிரி உள்ள ஒரு நியூ ஃப்ராக்மெண்ட்டை அதை ப்ரொடியூஸ் பண்ணியிருக்கு. Okay, can you understood? That method is known as the dispersive method. In conservative method, a same one double stranded DNA they can synthesize into the same another one double stranded DNA. That is known as the conservative replication. Then dispersive replication, that parent DNA molecule broken down into the fragment. Each fragment it is involving to produce in a new fragment. That is known as the dispersive method. Another one of the method is known as semi-conservative replication method. This semi-conservative replication method was proposed by Watson and Crick in 1953. This mechanism of replication is based on the DNA model. So they have given this method in the DNA model. DNA என்னது double helix model. அந்த மாடலை வச்சு எப்படி DNA it is involving in a replication process. They suggested that two polypeptide strands of DNA molecule unwind and start separating at one end. என்னது இதுதான் DNA molecule double helix structure. இது எப்படி இருக்கு coiled-ஆ இருக்கு. ஓகே, அந்த காயில்டாக இருக்கக்கூடிய இந்த டிஎன்ஏ மாலிகூல் ஒரு இடத்துல அன்விண்ட் அப்படியே காயில்டாக இல்லாமல் ரெண்டும் தனித்தனியாக பிரியுது. ஸோ இந்த பேரண்ட்லேருந்து வரக்கூடிய இது ஒரு ஸ்ட்ரக்சர், தென் இங்கேருந்து வரக்கூடிய இன்னொரு ஸ்டாண்ட். ஸோ த டூ ஸ்டாண்ட்ஸ் ஆர் போத் த எண்ட் they can separately. Okay, so they suggested that two polynucleotide strands of DNA molecule unwind and start separating at one end. இங்கே unwind ஆகி என்னது இது ஒரு எண்டில் இது ஒரு எண்டில் ப்ரொடியூஸ் ஆயிடுச்சு. ஓகே, டியூரிங் திஸ் ப்ராசஸ் கோவேலண்ட் ஹைட்ரஜன் பாண்ட்ஸ் ஆர் broken. இங்கே என்னது ஒவ்வொரு பாண்ட்ஸ் வந்து ப்ரெசன்ஸ் ஆயிருக்கு. இங்கே தைமின் ஆல்வேஸ் பாண்ட் வித் த அடினைன் வித் என்னது டபுள் பாண்ட். இங்கே என்னது 3 பாண்ட்ஸ் 3 ஹைட்ரஜன் பாண்ட்ஸ் வந்து ப்ரெசன்ஸ் ஆகிருக்கும் சைட்டோசினுக்கும் குவானைனுக்கும். எப்போ இங்கே அன்வின் நேச்சர் ஏற்படுதோ அப்போ இந்த பாண்டு வந்து broken down ஆகும். Can you understood? So during this process covalent hydrogen bonds are broken. The separated single strand then acts as template for the synthesis of a new strand. So இந்த தனியாக இருக்கக்கூடிய இந்த ஸ்டாண்ட் அப்படியே என்ன ஆரம்பிக்குது, it is start to produce in the new strand. Okay, subsequently each daughter double helix carries one polypeptide chain from the parent. Then this parent, single parent, they can produce the complementary another structure. So this is the new structure. So all the new daughter, this double helix, one strand with the parent strand, another one is the new strand. Can you understood? So each daughter double helix carries one polypeptide, starts from the parent molecule that acts as the template and the other strand is newly synthesized complementary to the parent strand. இங்கே parent strand எப்படி இருக்குதோ அதுக்கு தகுந்த மாதிரி complementary strand ஒரு new daughter molecules வந்து produce ஆயிருக்கு. Can you understood? So this is the daughter strand, this is the parent strand. So this method is known as the semi-conservative method. Can you understood all of you? Semi-conservative replication, DNA replication method. It is proposed by the scientist Watson and Crick in the year 1953. Okay, can you understood all of you? DNA replication. What is the meaning of replication? Multiplication. They can produce exact carbon copy of that DNA molecule. That process is known as the DNA replication. It is present in the three types: conservative replication, dispersive replication, semi-conservative replication. In a conservative replication, that original DNA molecule they can produce template at the same DNA molecule, the structure of a DNA molecule. That is known as the conservative method. Dispersive method. So இங்கே conservative methodல இதே மாதிரி இருக்கக்கூடிய DNA molecule they can produce the same another one newly. Okay, conservative. Then in a dispersive methodல இந்த DNA molecule என்ன செய்யுது, fragment என்ன செய்யுது, broken down ஆயிட்டு அந்த each fragment என்ன செய்யுது அதே மாதிரி உள்ள ஒரு carbon copies produce பண்ணுது. அப்போ ஒன்று வந்து என்னது old one, another one என்னது new one. So that type of replication is known as the dispersive replication. Another one of the method, it is known as a semi-conservative. In this semi-conservative method, that double helix model of DNA, then that unwind at one of the end with the broken of the hydrogen bond. One strand is the parent strand. Then the parent strand they can produce the complementary daughter molecule, daughter nuclei. Okay, into another strand they can produce. So that new daughter molecules that copies it is present, one is the parent, one strand is the parent, another one is the newly synthesized one. Okay, so that parent they can produce the exact of a complementary strand, another one new complementary strand. So this type of replication is known as the semi-conservative replication proposed by the scientist Watson and Crick in the year 1953. Okay, can you understood all of you? Okay, the next